That's good for now. And Davey, please just allow others to join as they join. So thank you everyone for being a part of this. And this is our first event of the TNT Industry Initiative. Today we're going to be diving into the next wave of neurotechnology, what's happening now, what's coming next, and how these innovations actually reach people. To set the stage, I wanted to talk about where neurotechnology is right now. Even a decade ago, what we've accomplished so far would have sounded unrealistic, but in the last 5 years alone, the FDA breakthrough device designations for neurotech have grown over 400%. The number of active BCI clinical trials have doubled since 2020, with more than 40 trials currently recruiting. And on the industry side, investment since 2019 has surpassed $8 billion. And so I think it's fair to say that this is no longer speculative, and neurotech is clearly transitioning from a what if and a research stage to deployment and having real impact in the world. And so we're very fortunate today to have four leaders and researchers at the center of this shift. These are people building the systems, algorithms, implants, delivery methods, and truly redefining what's possible. And so thank you all for joining. Let me go ahead and introduce our panel. So first off, we have Claire Baum. She is a postdoctoral researcher at Stanford working on the Artificial Retina project. Her work spans electrophysiology, neural encoding, and prosthetic design. I hope she will share more of her project now. Thank you, Claire. Please go ahead.
Okay, we're just launching right into it. Let's do it. Let me share my screen. Okay, quick recap. Thank you, Rahul. Quick recap on who am I? So, I'm Claire Baum, and so I'm actually secretly a physicist and got my BS in physics at UIUC, and then my PhD in the subdiscipline of atomic, molecular, and optical physics at University of Chicago. But then it was sometime in grad school where you kind of have to decide what comes next. And there was sort of a critical moment where I saw a video of a monkey playing Pong with its mind using the Neuralink BCI. And that triggered a cascade of events that caused me to take a hard left into, oops, sorry. Hello. See, a hard left into where I am now as a postdoc in Stanford, doing neuroscience and bionic vision. And so that leads me to the group I'm representing today, which is the Stanford Artificial Retina Project, which is all these lovely people. Um, and we are a grant-funded academic collaboration across multiple institutions. So University of Washington, University of Texas at Dallas, TU Delft in the Netherlands, and then Stanford, which is leading the charge in the Chicheldisky lab. And our goal is to restore naturalistic vision to people blinded by photoreceptor diseases. These are diseases like retinitis pigmentosa and age-related macular degeneration, using an artificial retina device, which is a type of bionic eye. And it's our approach to vision restoration, which sort of sets us apart from other vision restoration technologies. And our approach to restore vision is actually to restore the neural code. So what is the neural code of the retina? So here's a quick crash course in anatomy. This is the retina. It has three main layers of neurons. There's the photoreceptors, the interneurons, and the retinal ganglion cells, where the only light-sensitive cells in this whole retina are the photoreceptors. So when you see light enters the eye, goes back to the photoreceptors, which transduces the signal of light into electrochemical signals, which then effectively gets computed by the rest of the retina. And then these retinal ganglion cells send out little signals down their axons in the optic nerve. So these axons form the optic nerve bundle. And then the signals travel down to the brain and they give rise to vision. And it's these spikes in the optic nerve fibers from the retinal ganglion cells that we call the neural code. Now, in a retina that has been blinded by photoreceptor diseases, which kills the photoreceptors, this neural code becomes aberrant or maybe even absent. But in principle, if you were to restore every single spike, spike by spike and cell by cell, then perhaps you would be able to create an artificial visual percept that feels somewhat natural. And so this is the goal of the Stanford Artificial Retina Project. Um, Now, for decades, over decades, we've discovered that there are something like 20 plus different types of retinal ganglion cells that make up the neural code. and they each carry their own visual representation to the brain, and each in these complex patterns of spatiotemporal activity across the various types. But there's sort of a problem here, and it's that all the ganglion cell types are jumbled up in space. So if you were to introduce an electrode array and then stimulate according to some pixel map percept of some percept you wanted to evoke in the brain, um, you would be stimulating all of these retinal ganglion cells in the same exact way and not according to these unique encodings and unique patterns. So the percept you actually get is maybe not quite what you had intended. Um, but maybe in a weird situation, you would get this duck. But more realistically, it'd probably be this streaky pattern with blobs of light, and maybe it sort of resembles the percept you had wanted to evoke in the brain. So instead of doing this approach, we propose to introduce an artificial retina that can actually stimulate the retinal ganglion cells according to their specific cell types and with single cell resolution, which is important because they're all jumbled up in 3D space. Well, 2.5D. So this artificial retina will function in 3 stages. Stage 1 is that it's going to record the spontaneous activity of these RGCs so we can identify them and their types. And then we're going to stimulate and record to identify how each electrode activates each cell and their axons. And then we're going to stimulate optimally to represent an image and patterns of RGC activity. And it's pretty cool because we've already done feasibility trials of this in ex vivo experiments. And so now the game is to develop, fabricate, and test a tiny implantable device so we can do all of these things, but now in a living creature. So this is that device. And it's sort of a pancake stack of components that's 1.9 millimeters by 4.2 millimeters. And the first iteration of the device will be wired. At the heart of the device is actually a custom chip that we've made with our collaborators from TU Delft, and it contains 1,024 electrodes at 36 microns pitch, and each electrode is capable of both recording and stimulation. Some highlights of this device include that it can be inserted in a small incision in the eyeball. You can slide it through a little nail slot. It closely contours to the retina by way of this glass interposer, which we call the frisbee because it really looks like a frisbee, and that this electrically connects the electrodes on the chip down to the retinal surface. And then lastly, it has this grab feature on top that makes the whole thing maneuverable intraocularly by a retinal surgeon. So my role in this project as of right now is to oversee and also participate in the development of this device. And we're building it right now. But we have a plan for testing it after it is built. So this is sort of our testing roadmap. It starts ex vivo, then transitions to in vivo. Ex vivo number 1, we're going to test a simplified version of this device with isolated pieces of retina. These are like 2 by 2-ish millimeters living pieces of retina that we're going to put in an ex vivo setup that is sort of familiar to what we usually do. So we don't have to develop any new special techniques for testing this device electrically. If this works out, then we'll move on to an in vivo-like environment where we can both electrically and mechanically test the full device, and this will be put in a living pig eye cup. And then if this works, then we'll transition to in vivo, and we would test the full device in a living creature for the first time, specifically in a macaque monkey at the University of Washington National Primate Research Center Tissue Distribution Program. Ooh, mouthful. And so these will be in terminally anesthetized macaque monkeys, which means at the end of the experiment, the monkey will be euthanized. But these are monkeys that had been used for other scientific purposes in their lives, not just for us. And then lastly, if that works, ultimately the goal is to go to a human experiment, which would be likely a temporary experiment where we introduce the device into the eye of a person and probe whether or not stimulation according to this neural code actually does produce visual percepts that feel more natural. So this is the spiel. So thank you very much.
Thank you for sharing that, Claire. I mean, restoring vision is kind of a biblical problem, and it's so fascinating to see that it's actually being engineered its way into the world. Really, really cool. Thank you for that. And so now next we have Alessandro Ascani Orsini from Johns Hopkins. His work focuses on a miniaturized neuro device which integrates material science, wireless telemetry, and a nerve interface into an ultra-compact system designed for real-world deployment to detect deep TBI. Alessandro, can you please share more of your project? Thank you.
Yeah, of course. Hello, everybody. I'm Alessandro. I'm a third year PhD in electrical engineering here at Johns Hopkins. I work with Dr. Nitish Thakur, who I actually see among the audience. So that is great. And I'm going to be talking about the project that we've been doing together along with another lab, the Qiao lab. And this is the BrainSense project. So brain recovery assessment with innovative sensor engineering. So when someone has a trauma, any type of trauma, such as a fall or like a accident or like a stroke, the person goes unconscious, and we do not really know what is happening to that person, essentially. We do not know until we put the person into a CT or into an MRI machine, and that happens hours after the trauma. However, the first hour, especially right after the trauma, is the most important moment in which we want to monitor a brain injury, if there has been any. And we want to know, like, what is the triage level of this person? And can we do something to maybe prevent further damage, secondary damage of the brain injury, or to stop the brain injury maybe from spreading? And so what we're proposing is, what if we had, for example, a very tiny device that we could put in the ear of this unconscious person that could tell us, hey, this person has a brain injury, it should be prioritized over others, and that can maybe start an emergency protocol to then improve the likelihood that this person will recover from the trauma and that will not develop a brain injury, or that will essentially mitigate the damage from the brain injury. And so this kind of the overall encompass of the BrainSense project, but the main goal is essentially to build a portable emergency device that is able to diagnose, monitor, and improve the recovery from brain injury. And so we're doing this by looking mainly at two biomarkers. The first one, which is electrocardiogram through the ear canal, and the second one, which is instead is a new type of electrophysiological signal, which we call cortical temporal auricular activity. And this is a totally different activity that is picked up from the ear that essentially encompasses cortical activity, ocular activity, and muscle activity together, and tells us information about like the cortical and subcortical activity of the brain and state of the brain. And so this is the technology that Rahul mentioned earlier. We have a very novel sensor, which we call the Oris sensor, and is basically a soft, non-standard electrode that you put inside of the ear of the person. It has a proprietary e-textile that essentially covers it and is able to pick up those electrophysiological markers that I mentioned earlier. And this is, of course, soft, is scalable. We can build a lot of this, and then we can just put it inside of the ear and start recording instantly, basically. And then this is more the technology and the current state of our device. So we have, again, this is the electrode that you put in the ear. We have some miniature electronics, and basically all of this is then projected in the next coming few years to be miniaturized into an AirPod kind of device. And then we have this fancy connector that we use to connect the ear electrode to the device at the moment. So this is a translational talk, so I thought to go straight into the data. We built the device to monitor the activity, and we currently are deploying it within the clinic here at Johns Hopkins in the critical care department with Dr. Sungmin Cho. And we have been doing 10, 11 patients using this device. And these patients are critically injured patients that either had an accident or have been essentially diagnosed with a brain injury of some kind. So they received CT and MRI. These patients, as soon as they arrive in the hospital, they're put on what is called a continuous EEG, and they're put with chest ECG. And so what we have been doing is essentially using our device in conjunction with these other techniques to validate our diagnostic capabilities and our ability to detect acute brain injury or traumatic brain injury. So here in this case, we're showing, for example, our ability to pick up PQRS complexes. And so on the top, we have our signal taken from the device. On the bottom, we have the signal taken from chest ECG. And as you can see, the shapes match very well. And we have very high reliability within the device. We use heart rate essentially as one of the indicators for us to tell if there has been subcortical brain injury. And here is essentially a correlation between the activity taken from our device, which is the blue line, and the reference, which is essentially the activity from the EEG machine. And as you can see, they align pretty well, except for some spikes here and there due to data loss. But there's also a very good correlation, 0.94. So that's very high, and it's great. We're also able to pick up cortical activity or equivalence. In this case, we did a very simple, this was in healthy volunteers, by the way, very simple eyes open, eyes closed task. When you close your eyes, there is an increase in alpha power. Alpha power is the activity between 8 and 12 hertz. And so you can see that whenever the eyes were closed, there is a increase in power, which is highlighted by these red hotspots here. We also were able to monitor arousal, cortical arousal, using sleep. So we put the device in other healthy volunteers over a period of 6 hours while they were sleeping. And we were able to very well identify using our CTAA all the stages of sleep through which the person was going, starting from the beginning of the sleep with the eyes closed, then non-REM 1, non-REM 2, non-REM 3, and even REM activity, being able to see cortical spiking and sleep spindles as well from the ear. We have also been looking into applying a therapeutic aspect to it, even though this has only been done in animals. And so our idea is basically, okay, once we have detected there has been a brain injury in our patient, what can we do then? You know, we detect it maybe like 10 minutes from when the trauma has occurred. Can we do anything? And our idea is, well, very close to the ear canal, there is the vagus nerve that is passing by. So what if we attempt and stimulate that location? And we have observed that by doing this in cardiac arrest rodents, post-cardiac arrest rodents, we were able to essentially create and elicit a substantial change in both subcortical and cortical activity in the animal. And so this is a promising avenue for improving the recovery from a brain injury, for example, and essentially improve the likelihood that someone will survive maybe a stroke or an accident in the future. And so in conclusion, this is the whole view of our system. We have the ear location, we detect cortical activity, we detect cardiac activity and heart rate. We're able to, in this way, get activity from cortical and subcortical regions of the brain, determine if there has been a brain injury, and if a brain injury has been identified, we then start with a protocol, a stimulation protocol, to improve arousal and essentially improve the likelihood that the person will recover after the injury. And so this is, of course, a team effort. It's been a huge pleasure working in conjunction with Dr. Qiao and his whole team. And we really couldn't have made it without all these people that you're seeing on the screen. And of course, also our manufacturing partners that we're working with. So, yeah, that covers it.
Thank you, Alessandro. Very evident that Your work has a lot of scale possible and has a lot of real-world impact. So thank you for this. Next, joining us as well today here is Shubham Yadav from MIT, whose research on endovascular surgery-free brain implants recently achieved major traction, including a viral Nature Biotechnology paper, which is how I came across his work, and it demonstrated the feasibility of delivering neural interfaces through blood vessels. Shubham, congratulations on the impact and recognition of that work. If you could please share some more details.
Yeah, sure. Thank you for the kind introduction. Let me just share some slides and then I'll get started. Let me just, can you see the screen or wait, I think so.
Yes, we can see it. Yeah. Oh, not anymore. Not anymore.
Okay, perfect.
Yes.
So can you see it now?
Yes, we can see it. Yep.
Oh, perfect. Awesome. So, okay. So today I'll just talk about this work. I'm like circulatronics, and that's like part of like major part of my doctoral research at MIT, where we coined this term called circulatronics, which is basically like electronics circulating through the blood vessels. So that is like it's circulating inside the body, and then what we achieved with this technology was we were able to do surgery-free focal neuromodulation. And then for this, we developed these autonomous biohybrids, which can like say, like basically like they can like self-navigate themselves to the target region anywhere in the body. And then as a test case, we showed like focal neuromodulation in the brain so that we can show like if you can implant nanoelectronics in a surgery-free manner inside the brain, which where you have the most like difficult one of the most difficult barriers to cross, like the blood-brain barrier, then it's possible to actually achieve like surgery-free neuro, like say basically we can implant these hybrids anywhere in the body and then achieve local neuromodulation with those technology. So before I give you like say like I want to give you some background about why do we need this kind of a technology, or like say what is the state of the art ongoing in the field of neuromodulation. So to talk about that, so yeah, current state of neuromodulation technology, something like we can basically divide the field of neuromodulation into two types. One of them is like say non-invasive brain stimulation technologies. So this includes technologies such as TMS, TES, TFUS, and a few more. So the nice thing about this kind of like technologies which have existed for decades now is like these are non-invasive, so you can readily use them for clinical translation, like the clinical work, like translate it into different applications and therapeutics. But they have a major fundamental drawback, which is like they cannot achieve focal brain modulation. I'm like, if you look here in this graph, we can see I'm like the focality, the spatial resolution with these technologies, it's on the order of millimeters and more. But when it comes to therapeutics, or like when we are talking about specific diseases, they need resolution which is like on the order of hundreds of micrometers or lower. So fundamentally, these technologies just cannot achieve focal neuromodulation just because the way this technology works in general. The good news is people have been working with focal like modalities as well. I'm like, however, the I'm like this includes technologies such as deep brain stimulation, and then there are like say newer novel wireless electronic devices which have been able to achieve focal neuromodulation. But the major challenge with all these technologies is that you have to basically do a intracranial surgery, which involves some like you have to like basically drill open a hole in the skull and then implant the electrodes. I'm like, when we talk about, let's say, deep brain stimulation as such, I'm like, here, if you see, I'm like, these are some of the, like, say, images taken from clinical trials, like with DBS, or like this one is like, again, like some human patients which have undergone these surgeries. I'm like, here you can see, I'm like, the surgeries are really invasive. And then on top of it, there is a high risk of like infection, like surgical infections, device failure. And then any of these risks when you have like say infection happening or like the devices when they fail, you are basically talking about you have to retract the devices back and then you have to implant them again. And when you do the implantation again, we are talking about going through these similar procedures every time we have to do this. So it's not an ideal situation. And then when it comes to, let's say, newer class of nanoelectronic devices, where researchers are now making them smaller and miniaturized, I think as Claire and Alessandro have already talked about different kinds of devices they have been working on. So here also the idea is the same. I'm like they are miniaturizing it, but no matter what we do, you still need to do a intracranial surgery. You need to like open up the skull, like, and then implant things into the brain tissue. So the major drawback, or like the fundamental drawback of these focal, like say the advanced or the current is like current neuromodulation technologies, is still that they require an invasive surgery, and then it has its own drawbacks. So to talk about some of the drawbacks, I mean, these are just a few, like which are the most important, but then there are like n number of more drawbacks which comes with it. So it's like when you do a brain surgery, I'm like there is a 4 to 9% infection risk. And then the other thing is, I'm like there are so many patients who need these brain, like say who need like neuromodulation therapeutics, but just because of the invasiveness of these technologies, only 1% of the patients are eligible for these kind of therapeutics. So it's like the current state like of technologies is that there is like so many patients out there whom like even they need a like therapy, like a neuromodulation therapy, but like they're just not eligible because of like say the patient history, and then the kind of invasiveness which might be associated with the treatment they needed. So it's just like we are almost like we are ignoring 99% of those patient population. And more importantly, I'm like, there are so many different regions. I'm like, thalamic nuclei is just one of them. The other one is this deep G. That these are like, say, just a few like brain regions which we are talking, we can think of like where you cannot even think of injecting or like say doing a surgery in those specific regions because they're like so critical for functionality that these these regions are pretty much considered as touch me not when it comes to like the human population. So it's like there are so many things which we are missing out just because the way the current neuromodulation technologies are being developed or being approached at. So we asked ourselves a simple question, which was like, can we achieve focal brain stimulation in a surgery-free manner? Because I'm like, when it comes to bioelectronic, like say implants, we can already see I'm like most of these technologies they leverage like say like how should I say it? I'm like they leverage the development in the nanoelectronic paradigm where I'm like people know I'm like there are so many new kind of devices which are coming up. So we know the benefits of these electronic devices, but the major challenge is the invasiveness or like making them like say getting them to be implanted in the target regions in a safe manner. So the idea was, can we achieve this surgery-free bioelectronic implantation, and we can do like brain stimulation based on that. So for this, I'm like we had a, like a slightly different inspiration. So I'm like when we look at our human body, we know that the entire body like has a circulatory system where the blood vessels reach each and every part of the body from like brain to all the way to like say peripheral nervous system to other major organs in the body. So it's like we have a circulatory system which penetrates everywhere in the body. And then when it comes to like say the agents, I'm like what exists in all of these, like say in the circulatory system, we know that we have immune cells which can actually like not just immune cells, like basically like say we have like all different kind of cells which like have their specific function, and then they can actually reach each and every part of the body under certain conditions. I'm like, red blood cells are easy. They carry, like, say, the oxygenated blood. I'm like, they help us like circulate energy. It's easy. But when it comes to, let's say, like when we have some kind of a disease, the first responders in the body, we already have something like the immune cells. I'm like, there can be monocytes, there are macrophages, there are T cells, there are like B cells, there are like say NTK cells. So I'm like there are so many different kind of cells which can be the like say are agents which can reach each and every part of the body in case something goes wrong with the body. So what we thought is like say, hey, like we already have a circulatory system, we have different kind of cells which can already reach different part of the brain. So how about we miniaturize the nanoelectronics, that we make technologies which can be like really small, and then we make something like a hybrid of a cell and electronics, so that when the body is in a disease stage, when there is a target region somewhere in the body, these hybrids can actually reach there. And then we do not need to do a surgery, but just an injection in the vasculature. So it just like becomes like a vaccine for neuromodulation rather than doing a surgery and then making it invasive and the associated issues which comes with it. So this is what we envision. And then that's where we started working on it. We developed these like cell electronic hybrids, and then we called this technology circulatronics because now we have nanoelectronics circulating in the bloodstream. And then I'm like, this is just an schematic. So here you can see, I'm like, when you have an inflammation somewhere in the brain, let's say, so this is the region where like the inflammation is happening, and then these hybrids are going through the vasculature, and through the diabetes process, these hybrids can actually cross the blood vessels, and then they are just homing towards the inflammation because I'm like, when we're using these immune cells, I mean, we used monocytes in particular, and then monocytes are known to because of their like chemotaxic properties, they can navigate themselves or reach the inflammation regions like just by themselves. So it's like now we have a autonomous system where if there is inflammation anywhere, let's say in any part of the brain, the monocytes will find it, they will reach there, and while they are reaching there, they are carrying the nanoelectronics along with them. And then once we have the electronics, so the idea is we are making these nanoelectronics to be wirelessly activated. So you can actually activate these nanoelectronic devices from outside the brain in a transcranial way, so that we do not need to implant an energy source. The idea is I'm like, you want the entire thing to be surgery free. So it's like you cannot have wires with the electronics or a modality where you need to like do some surgeries to deliver energy to these devices to be activated. So what we did is like we used optoelectronic devices, created these hybrids with of monocytes and optoelectronic devices, injected them in the blood vessels, and then the monocytes autonomously cross the blood-brain barrier, identify the target region, get them self-implanted at the target region, and once the devices with the hybrids are at the target region, we use say NIR light to to activate the optoelectronic devices and then do local like local neuromodulation with those devices. So I'm not going to go into the details of like everything because I'm like, most of the details are in the paper and like it's online now. So I'm like, it took us a while to get it out, but it's finally out there. So I'll just like summarize some of the experimental results which we got like during part of this work. So I'm like, we developed these optoelectronic devices. And these are basically photovoltaic devices, three-layer structure. These are 10 micrometer in size, 300 nanometer thin, so really flexible and highly efficient. I mean, we developed the highest efficiency photovoltaic devices at subcellular scale. I mean, these are better than the previous reported thing by orders of magnitude. And then we were able to, we were just testing out whether these devices are effective for neuromodulation or not. So we are actually able to do bidirectional modulation. So I'm like, when people talk about neuromodulation, the first thing which comes to everyone's head is, does it stimulate? Or basically like, do you make neurons fire like at higher action, like does it evoke action, like can you make neurons evoke action potential? But we can not only make neurons fire faster, we can make the activity go down as well. So we can actually do like neural inhibition as well. So I'm like, this is not published yet. So I'm like, but it's like the work is under review, so we'll have this out as well at some point of time. But we can do bidirectional neuromodulation control using these devices. And then we created, so when I'm talking about these hybrids, so this is how the hybrid actually looks like. I'm like, this is a Fabaceum image of the monocyte carrying the photovoltaic device on the outer membrane of the surface. So this is the monocyte device, and then you have this photovoltaic device attached to it. So you can see how thin this layer is. I mean, this is like say around 300 nanometer thin, and then a monocyte typically is around 10 to 20 micrometer in size. And then we created these hybrids. And then when we do like this surgical neuromodulation, we were recording using like single unit electrophysiology. And then we were able to actually achieve a resolution of 30 micrometer, like a spatial resolution of 30 micrometer with this technology. And yeah, of course, it needed a lot of people's effort and work which went into it. Of course, Deblin, I'm like my PI, I'm like my PhD advisor. I'm like nothing would have been possible without her. We really had like so many discussions. It's years of work where we like going back and forth in terms of the design rules or design principles, what kind of devices to work with, what kind of cells to use. And then when they're like Benoit, Martha, these are like the postdocs who were there in the lab at that time, and then they helped a lot with the device design. Ray and Preet, these guys helped a lot with the animal side or like say the device side as well, and then the entire NCB team and like everyone chipped in at some point of time. And then a lot of the work was done at Koch Institute, MIT.Nano, that MIT CEHS, and then we had a lot of funding coming from NIH, Mathers Foundation, and like Mind Prize as well. So a lot of funding agencies helped us achieve this. So we're really happy that we're able to demonstrate something like this. So yeah, I'm like, I'll just stop sharing.
Thank you, Shubham. I mean, truly fascinating work. It really adds to this theme of engineering sci-fi concepts into the real world. So congrats on all your success and much more to come. And so last but definitely not least, we have Shaoyu Qiao, Director of Neuroscience at Paradromics, one of the leading high-bandwidth BCI companies pushing towards scalable clinical systems. Shaoyu, congratulations to you and the Paradromics team on receiving FDA clearance recently to initiate your first in-human clinical trials. A huge milestone for the field and a lot of impact and scale ahead. So if you wouldn't mind sharing some of your work as well, thank you.
Right. Thank you so much for the invitation. Very glad to meet you guys all. I just want to save more time for trainees to ask questions. But as you said, Matt Engel, our CEO, founded this company 10 years ago. So this is really a big milestone for us. So I joined the company 4.5 years ago, really overseeing this preclinical testing to clinical translation. So a lot of hard work and then really experience how we can translate the novel technologies from the lab to clinic. So that you requires another side of team that help you really truly understand how to build a medical device. So meaning you need the quality team, you need clinical team, you need regular team, you need manufacturing. So not just scientists and engineers. So that's really the truly how I can say translational work. So we are very pleased we get this good news, but a long way to go, right? So we still need to continue to work hard and then learn from the field. As you can see, there's so many fascinating technologies emerging at this point. So hope we can build a good. So we are establishing a new field. So neurotech is a new field. I hope we can collaborate in the future very soon. So.
Thank you. Yes, and I think as a field we are growing, and the more collaboration, the better, and the more benefit we could derive to our patients and people. So thank you everyone for your overviews. We touched on some great points, looked at a lot of real-world applications. I do want to have sort of a brief discussion at this point. We are pushing a little bit over time, but no worries. I wanted to ask the guest panel, firstly, for translational hurdles. What are some of the things along the way, either technical, regulatory, logistical, clinical, whatever it is, what has really stood out to you and has served as a significant hurdle? Thanks.
I can take that probably first because I experienced that. So I'm just like you guys, like a scientist and engineer trained in neuroengineering field. And then I got into this opportunity to really go through this journey. I think it requires a cross-functional team. Definitely understand what's your clinical needs and what's your design rationale. What kind of things you want to build to help patients? You definitely need a quality regulatory and clinical manufacturing team to work together with scientists and engineers to go through this whole process, right? So if you are interested, you should take a look at all the ISO standard that we have to follow, all kinds of preclinical testing protocols we have to go through to show FDA the device or the new drug or whatever you want to bring to the clinic that's actually safe, right? And then that's working. So that needs, no one can do that by themselves. You need build a cross-functional team. That's what I learned.
Right. Thank you, Shaoyu. Yeah, and
Yeah.
definitely immense scale and pathways for developing these out into the real world. So thank you for touching on that.
Yeah.
I think I'll
ne-
riff off of that maybe.
yes, amazing. Thank you.
So definitely you need a cross-disciplinary team. I think one of our struggles that we've been having is that we are an academic lab, but we're sort of pretending to be a startup. So we have the funds of an academic lab, we have the personnel of an academic lab, and it is not nearly that of something that is like VC funded. And so there are grad students who are aboard that are looking to get PhDs, and they're still in a phase of training. But it seems like our projects are shifting more and more toward engineering projects that are no longer really PhD projects. So we're caught in this like weird in between academia and industry, where we're like both and neither at the same time, and we need more money, like always, and more personnel. So it's been really trying to balance progress moving forward while also trying to like consider what that future will look like. Will it look like we spin out into a VC funded company? Will it be a nonprofit? Will it be something called a focused research organization, which is an FRO? That one's sort of my favorite. And then what does even like the path to human look like? Because we need to show many different steps on the way to human, and safety being one of them, which is like a big one. And then even when you get to a human, does your technology work? So yeah, and good technology is sort of the foundation of moving forward beyond that. So there are lots of little gotchas in the process of developing technology, and we're still decently far out from human, but that's the dream.
Yep. So I can actually follow up kind of on that. Well, like going ahead and translating this device, in our case, the auricular device, it you really need to take a step from academia and start actually focusing on like more like everyday engineering problems that might not be as innovative, but might be necessary for making the device reliable, repeatable, reusable. And I actually want to add on that another thing that I think is, at least I learned, is to really make your research and your work very like intuitive and I think of how Apple made like the computer, like the very nice, sexy computers that we use. And like we kind of need to do the same also with our tech and our pitches, because I mean, at this presentation, I saw some beautiful figures and I understood them instantly. I think that is very important, especially in these cross-disciplinary environments, because you're talking with clinicians that know nothing about engineering, and you're talking with engineers that know nothing about the clinic. So communication is extremely, extremely important, and the intuitiveness of your device, of your application is key, essentially.
Maybe I'll just add on the scalability part of it, because this is something when we were developing this technology, I'm like, I didn't show one image, but I'm like, when we make these devices, I'm like, so basically, I'm like, to do one round of experiment with animals, we need around 10 million of the devices. So the thing is, if you're talking about order of millions, I'm like scalability, like not just like the device, like say perform. I'm like, it's just not about like you have to make those many things, but you have to make it reliably, and you have to develop processes which are scalable as well. So I'm like, this is was literally one of my nightmares. I'm like, I struggled for more than a year, where I'm like, we were making everything CMOS compatible, making sure things are going in a scalable way. But when we were collecting these samples, or like say sorting them out into like a specific hybrid, like which has a certain like a device attached to it for sure, it was not easy. I'm like, it took us a lot of time. And now we look at the same problem now, because I'm like, we want to take it to like say clinical trials at some point of time. But when it's a small mice, you need, let's say, 10 million of these hybrids to show some functionality. But you scale up the body from a mouse to a human, you're talking about, I don't know, maybe like close to 100 million, or like maybe a few 100 million of these hybrids. So the scalability of it, let's say, like we still don't know what are the right answers, but I'm like, when people are developing these technologies, they need to think of how you can scale things in a nice, efficient way. Because for us, I'm like, if we just are going to take 10 or like say 100 times more time, like say the if like say the complexities are just linear, the numbers don't make sense for us at all. So it's like it's going to be a big challenge, which we are still figuring it out how we can achieve this. But in general, I'm like, when people are like, I'm like, if you're doing let's say clinical trials, I'm like, the technology needs to work, but it needs to work in a scalable way. So that part I feel like is really important while you are like say, initially just in the prototype or the design phase, to understand, okay, this is the vision for this technology, and then how we go about building those things. So.
Thank you. Yes, it's very evident that these technologies have very long timelines and explicit pathways into bringing this into translation. So thank you everyone for sharing a little bit of that perspective. I also wanted to ask and follow up in regards to this is, you've been developing these technologies for a while. What is one big lesson that you've learned while working on your project? There are a lot of graduate students in this call, maybe in the midst of their projects or at the beginning of the project, sort of something you can share to help out the next wave. Thank you.
Maybe I can go first. I'm like, it's like the most recent like work. So the thing is, I'm like, one thing I realized, I'm like, this is so I'm trained like I did my undergrad in electrical engineering, and then for my PhD, I'm like, I think as Claire also like moved from like for a postdoc into like interdisciplinary work. I'm like, it was the same for me, where I'm like, I used to do modeling of semiconducting devices, and then cut to PhD, where I was doing experimental device physics, and then on top of it doing a lot of animal work and neuroscience things. So sometimes I feel like when you're doing like working on complex problems, they are complex, and then you have to acknowledge it at some point of time that science is hard, it's really difficult, and sometimes it's okay when things are hard. I'm like, like the thing is, when people say, okay, you should find the easier or like the more optimal way out of the things, but most of the time it just doesn't work. I'm like, if things are difficult, you acknowledge that it's difficult, and then keeping that thing in mind, at least your perspective changes a bit, and then you can actually make slowly, and you can make progress over time in a slowly but a steady way. So I'm like, that was my biggest learning, that sometimes it's just so difficult to do research or like do like make something which actually works, and it's okay if it takes time, because good things always take
I want to say, in my case, I learned I think it's very important to have a big team, as like a very essentially like multidisciplinary team, so that not everything falls on you and you can delegate stuff down the line. You know, there's the person that does the clinic, there's the person that does the data analysis, there's the person that like refines the device, and I am very grateful about all those people, because otherwise I would be doing that all myself and I would hate it and I would get nothing done. So yeah, make sure you have a big team and like, yeah, rely on them a lot. Yeah.
I totally agree with what both Alessandro and Shubham just said. Yeah, collaborate, collaborate, collaborate. And then, yes, things will go wrong. It's not a matter of if, it's a matter of when. um, I think I also want to add maybe a couple lessons learned is that I think it's very useful to sort of sketch out a path to the finish line before you even begin on a project. So you don't have to have everything figured out, but if you assume step 7 of 10 will work by magic, then maybe you better question whether that's the right path or not, because then when you get to step 7, it could be that you've wasted a lot of time because actually step 7 just doesn't work. So having a path to the finish line that's at least plausible, I think, is important before you even take that first step. And then secondly, time is money. Sometimes it's better just to buy the thing than say, make it yourself. I see Shubham laughing. I will conclude with that, I think.
Amazing. Shaoyu, do you have any thoughts?
I mean, I have so many thoughts, but I don't know which one I should. Yeah, so I'm gonna piggyback from Clara. So I think for the core capabilities you want to build, you would like to bring in-house. So that can actually shorten the cycles, because this is you try something new and then you want to learn the results of that. So the core capabilities is better to bring in-house. But everyone said we need a bigger team, but bigger team's not for free. You need money, you need funding to build a bigger team. So that's a conflict, always the case. So we always work in this like resource limited environment. And then, so the key is how you can balance this out. That's a tall task. So that requires the entire team to get aligned. Communication is definitely the key. So I totally agree with that. So yeah, I'm happy to share more offline, but yeah.
Thank you. Yes. So we are nearing the end of an hour. I do want to leave a little bit of room for any audience questions, so I will open the floor a little bit. If you want to share on chat or even just unmute your mic and direct your question.
Hi, I have a question.
Yep, you can go ahead.
Yeah, first I want to thanks every panelist for an amazing talk you gave us, and that's very helpful. And then I have one specific question for Xiaoyu. So what's the one skills that you feel like your academic training prepare yourself the most when you transfer yourself into an industrial position? And then what is that one specific skills that you feel like, okay, I didn't see it happening, and then I have to learn it throughout the way I spend time really in the industrial role and then perspective.
Work ethic. You need to work hard, and then pursuing hard problems, dig deeper, because a lot of skills you actually can learn, but we would like to hire someone actually dedicated to the work, working hard. Yeah. Yep.
Thank you.
Yeah. No shortcut, no curveballs. Working hard. Yeah.
Thanks.
Okay, it seems we have no more audience questions, so I do want to end with like one optimistic tone, and I do want to ask the panel, what do you envision for the future of this? We're at the start of it, we're at the precipice of something that's great and is going to impact a lot of lives and benefit a lot of humans. What do you see coming for the future? What are you excited about? If we could get anyone to share their thoughts on that, it'd be great.
Let's say future is bright. As you can see, there's so many new ideas, right? We would like to see, I mean, we can already guarantee all approaches going to work eventually in clinic, but there's so many health problems we can solve. So that requires different ways to solve that problem. So the key is like, as you said, we need to keep this positive. So the journey going to be tough, but I think future is bright. So that requires teamwork, requires new ideas. So that's why we actually getting trained right now to be the next generation engineers and scientists to push the envelope. So a brain or nervous system is so complicated, so no one knows how that works. So that requires generations efforts to bring new technology to help patients. Yeah.
Okay, I'll go. Regarding the BrainSense device, our goal in the long term and what we're really excited about is really making this technology essentially be the all ICU just in your pocket kind of thing. So essentially having this device that you put in the ear and does a full diagnosis of the brain, and it's non-invasive, it's wearable, and that's what we're trying to give in the future. That's what we're excited about. I mean, and now we're already bringing it in humans. It would be great if we saw it in ambulances and being routinely used in hospitals. Yeah.
Yeah, I'm super hyped for the future for both like all neurotech and our particular project. I think something I wanted to comment on was the science aspect. Is we're building all these tools for neurotech, which is profound for doing things for people, but it also sort of carries along the science. So with our particular device, we're trying to restore vision, but it also leverages the science aspect. So by trying to improve vision, you're sort of learning how does the brain work inform vision? What happens when you stimulate one cell in the retina? What percept does that lead to? What if you do two? What if you do three? What if you mix up different types and like change their times in terms of their relative stimulation? You're literally trying to figure out how is vision built in the brain cell by cell. And I think that's just super cool and will be very profound and will hopefully sort of paint the future of how maybe BCIs will lead to higher resolution percepts more so than these sort of coarse-grained things today.
I can just add one more thing. I'm like the nice thing, I'm like the future is so bright. I like it because I'm like a majority of the research which was going on in the academic labs and like in the past, it was not getting translated into real world like an applications. But over the last few years, I'm like we see increasingly more and more research labs, like academic labs as well. They've been focusing on translational work, which is really amazing. I'm like even for us, I'm like this paper, I'm like will be out there, but I'm like we have already started, like say like we all also have like one of the starters, which is like say focusing on taking this work to the to translational work. So I'm like really hopeful that I'm like more and more academic labs, they do interesting, really like nice research, and then they bring it to the clinics, to the translational work, and then we get more and more technologies working in real life. So yeah.
Amazing. So that marks the end of the hour. TNT and Hopkins would like to extend a real heartfelt thank you to all the incredible guests for sharing their discussion and their insights that bridge science, engineering, real-world translation. I'm sure we all learned a lot. Everyone in the audience, thank you for being here. This session marks the first event of the TNT Industry Initiative. We're excited to say this is the beginning, not the end. We'll have many more industry and research-facing events in the coming semester, and so please keep an eye out for upcoming announcements. Thank you again, everyone, for being here and for making this launch such a strong start. Thank you, everyone.
Great.
And thank you, Rahul.
Thank you.
Thank you. Bye.